DNA can be found in various forms. The first form that you saw on the previous slide was that of chromatin. Chromatin is condensed chromosomes, and this condensed state is very important because it helps to protect the fragile chromatin threads during cell division. So looking at our slide, we can see the double helix, which you were introduced into in chapter 2. The DNA surrounds protein particles called histones, and the chromatin can be thought of as beads on a string, a structure with a nucleosome. So the nucleosome is 8 histone proteins wrapped by 2 lines of the DNA double helix. On the right-hand side of the slide here, we can see chromatids, and the chromatids are part of a chromosome. So this would be one chromosome. This would be another chromosome. And what we're looking at here is a metaphase chromosome, which is at the midpoint of cell division, and it consists of 2 sister chromatids. So our slide here is showing us the cell cycle, and the cell cycle is showing us the parts of cell division. So we can see that the majority of the cell cycle is including what's called interphase. So G1S and G2 all make up interphase. And even though the prefix might remind you of intermission, that's not what's happening during interphase. It's an extremely active phase, and it's a time period when there is DNA synthesis that is occurring. There's the production of new enzymes, new organelles, and it's the preparation for mitosis. So if the proper preparation for mitosis does not occur, then the cell will destroy itself, which is a really important thing to prevent something like cancer from occurring. So what you see here are these important checkpoints. We have a G1 checkpoint, a G2M checkpoint. You don't need to know the checkpoints, but what they are is basically a time frame when it can just double check to make sure that these things have occurred properly. If they haven't occurred properly, then the cell destroys itself. Also, you don't need to know these specific names like G1S and G2, but you should know that DNA synthesis happens during interphase, final preparations happen, and these events occur as well. So a cell that is in interphase is going to look like what we see up here. The nucleus is still going to be intact. There are going to be 2 centrioles together form the centrosome matrix, and then some of those things will start to change during mitosis, because mitosis is the division of the nuclear material, which is the most important material of the cell. You'll need to know the 4 different phases of mitosis, followed by cytokinesis. And you'll need to be able to identify them for lab and know what's happening during each phase for lecture. So for example, this phase here is metaphase because all of the chromosomal material is lined up in the middle of the nucleus. This phase on the left is telophase, and we know that because we almost have 2 full new daughter cells, And there is the presence of what's called the cleavage furrow, which is the point at which there's an invagination of the cytoplasm and it starts to separate into two new daughter cells. So our next two slides are going to show these slides, these phases in a little more depth. So mitosis is the process of nuclear division, which chromosomes are distributed to two new daughter cells. After these four phases occurs, cytokinesis happens, and that's the division of the cytoplasm. So during interphase here, this is what we would see. We would see centrosomes with centriole pairs. The nucleolus is inside of the nucleus. The nuclear envelope is intact, and the DNA is in the form of chromatin. So it's a period of cell activity. The DNA, as I said, is in chromatin, and there's distinct periods. During prophase, and you don't need to know the difference between early and late, first what's called a mitotic spindle forms. And the mitotic spindle is an organizing structure because it organizes the DNA. The centrioles are going to start to spread out, and there's what's called an aster that forms. So the chromatin coils and condenses, forming chromosomes. Each duplicated chromosomes has identical threads called sister chromatids held together at a centromere. And as the chromosomes appear, the nucleoli start to disappear, and the two centromeres separate, or centrosomes separate from one another. The centrosomes become a focal point. They start to form this microtubule assembly called a mitotic spindle. And those microtubule arrays are called an aster. Later on in prophase, the nuclear envelope starts to break up, allowing it to interact with the chromosomes. And the remaining spindle microtubules now are going to be where the chromosomes kind of scatter to. And we have what's called an equator of the cell that forms. So you can see, first of all, that the nuclear envelope has disappeared. So there's fragments that are left. The centromere is where the two chromatids meet. So this centrosome has two sister chromatids. The next phases of mitosis are on this slide. So after prophase, we have metaphase, and metaphase means middle. So we have this mitotic spindle. Again, it's organizing structure where the chromosomes are going to be attached to. They line up along the equator of the cell, also called the metaphase plate. So the two centrosomes are at opposite poles, called the equator or the metaphase plate. After metaphase, anaphase happens, and anaphase is the shortest phase of mitosis. What's going to happen is that the moving chromosomes look V-shaped, and they're going to start to be pulled towards each daughter cell. When we can start to see the beginning of the daughter cells, we have a contractile ring that forms at the cleavage furrow. And a lot of this is the opposite of prophase because now the nuclear envelope forms as well as the nucleolus. So it begins as soon as the chromosome movement stops. It's kind of like prophase in reverse. A new nuclear envelope forms. Mitosis is now ended. Then after telophase is cytokinesis, which is the division of the cytoplasm. It can begin during late anaphase or telophase, and the actin myofilaments form this cleavage furrow that pinches the cell apart.